Hello, everyone. Good morning, good afternoon, good evening. Marc Estigarribia here again, Managing Director of MSQ Ventures. We are honored to host Dr. Catharina Svanborg, Founder and Chairman of the Board of Hamlet Biopharma, to our MSQ CEO Founder webinar series. Hamlet Biopharma, the innovative pharmaceutical company currently advancing several phase 2 trials targeting infectious diseases and cancer, along with an extensive project and patent portfolio. Professor Catharina Svanborg, how are you today?
Fine, thank you. Thank you, Mark, for the invitation.
Great to see you again. MSQ is based in New York City. Our audience is global, spanning 20+ countries. We have strong presence in China. Good evening, Shanghai. Where are you calling from, Dr. Svanborg?
I'm calling from Lund, Sweden, where our group, scientific group, is based, and also our company.
Great. It's getting a little chilly in New York. What about where you are? Is winter upon us?
Still mostly rainy, but just a few miles north of here, there's plenty of snow.
Okay, fantastic. You know, as a founder of Hamlet, we're very interested to hear, you know, what the motivation and just the journey. If you can share a little bit about your inspirational journey and founding Hamlet, we would appreciate it.
Thinking about this question, it occurred to me that I come from a family of medical doctors and entrepreneurs, and working with science and making discoveries with my group, we realized, as we found these golden nuggets, these molecules with very special effects, that it was almost an obligation to take the academic research, commercialize it, and try to get it to the people who need new treatments. And this is what we're doing. It's also a very exciting lifestyle to have both live with the scientific part and the commercial development.
Great. I know that we'll probably be learning a little more when you share your company presentation, but just at a high level, where are we today at Hamlet, and you know what should we be looking forward to in 2024 with milestones over the next 6 to 9 months? Just high level.
So we are in phase 2 trials with 3 different projects. And especially for the bladder cancer project, we are very excited because we just received fast track designation from the FDA. So there we're seeing hopefully a rapid progress to phase 3 trials together with the FDA advisors. The other 2 trials are in bladder pain and also acute cystitis, which are huge markets and unmet medical needs. And we are seeing some very exciting clinical data, hopefully coming out of those 2 studies during the next year.
How did Hamlet decide to focus on brain cancer and bladder cancer? What was the driver?
The driver, as many things we do, the driver is what we find. The discoveries, the science shows us what is possible and what are good questions to ask. So bladder cancer, we were working with an animal model that showed us extremely good protective data when we just injected Hamlet into the bladder. Brain tumors, a collaboration with a leading Norwegian group who had an animal model for human brain tumors that we tested, and it worked beautifully. And finally, colon cancer, which is another interesting area where Hamlet showed very potent effects in a human-related animal model. So this is really why we made the choice. The clinical choice of starting with bladder cancer, of course, depended on our clinical colleagues. You know, the collaborations with experienced urologists in Sweden and in Czechoslovakia at that time, who saw the potential and were willing to help us along with the process.
Great. Can you tell us a little bit more about your management team in terms of, I always surround myself with a lot smarter people to help me with what we're doing here at MSQ. Can you tell us a little bit more about your management team in terms of assembling your team?
Yes, there is a strong philosophy, which your question is exciting for me because I get an opportunity to explain how can we have, many people are wondering how we can have scientific excellence and the company in the same group of people. And this is made possible by very extensive collaborations with experts in different parts of the fields that we are touching on. So during 1 week, there may be about 40 different individuals who are involved in the work. There may be production ongoing, the clinical trials, the patent agencies, the US team who are helping us with the FDA, and then very much the core team, our platform, drug development platform that we run with both science, discovery, and all the different things that are required to fulfill the legal requirements and other things.
Great.
So it's a flexible team. It's probably also very cheap.
Yeah, of course. Going we're towards the year end. 2024 is upon us. What are the exciting things that we're looking forward to in 2024?
For us, the fast track, which came late in 2023, gives us a taste of what is possible.
Mm-hmm.
And so we're hoping that the strength of the bladder cancer project is also going to be reflected in the other 2 trials that are ongoing. There are huge populations out there. Bladder cancer is a huge indication, and so is acute cystitis. And if we can manage to take our products to the market, we will help a lot of individuals, and I think the investors will also be very happy.
Great. I guess you know, a lot of companies strive to accomplish what you have accomplished. Any advice you can provide in terms of you know, all of the milestones that you've hit? Are there any sort of anything that you can share to help companies through their hurdles and opportunities and focus, discipline from your accomplishments?
Yes, I don't want to be presumptuous in this regard because there are obviously experienced big pharma individuals with extensive experience on a completely different level. But I think what we're trying to do is to let the bottom-up creative part of the organization maintain a significant grip over what we do. Of course, there is top-down. There are all the structures that must be in place for a formal drug development, but we try to maintain as much as we can of creativity and or of the individuals that work, the smart individuals that work with the team. And maybe that's partly why we have been able to do this. I think so. I'm grateful to them for having hung in there and kept us going in the way they have.
Great. Well, thank you very much. Well, we're looking forward to the knowledge share of Hamlet's novel approach. We'll take questions from the audience in the back of your presentation. So Professor Catharina Svanborg, please share your screen and start your company discussion.
Thank you very much.
Great.
So it's a great pleasure. And again, thank you, Mark, for giving us the chance to present Hamlet Biopharma. Hamlet Biopharma is an innovative pharmaceutical company listed on Spotlight stock market in Stockholm, Sweden. We are developing groundbreaking scientific discoveries to the clinic in 2 areas, cancer and infection, and have built, as I described, an organization where both the discovery mode can be on and the drug development mode at the same time, and the individuals can use their talents to develop both sides of the coin. The successful approach was recently shown in a clinical update where we analyzed data of studies in patients with bladder cancer of our lead compound alpha-1H. And the studies showed excellent outcomes. And as I said, we have received fast track status based on this data from the FDA. Hamlet Biopharma was recently formed by a strategic merger of initially two biotech companies, one in the area of infection and one in cancer. And we find that this merger is giving us the muscles and also the product portfolio that is required to take this work further. So the drug development side is benefiting from a broader project portfolio and, of course, lower risk for the investors. The patents, a rich patent portfolio, 15 patent families and 60 individual patents. And also publications. Our group has a very strong track record for publications, over 500 papers in international journals, and we are ranked in the top 1% in the world with the Stanford ranking system. So, treatment of cancer and infections improve the lives of many. At least that's what we hope. The merger was essential, as I said, because it created a company with a larger project portfolio and also improved commercial potential. The work is done through, in part, an agreement with the technical development platform, the one that I described as merging discovery with drug development. The projects and patent portfolios are about alpha-1 for bladder cancer, the receptor antagonist for bladder pain, and IL-1 receptor antagonist for recurrent cystitis. So the 15 patent families, where 8 are for cancer treatment, 7 for infection, and 1 for tuberculosis. So collectively over 60 individual patents. The slim and flexible commercial organization, I think Mark's question already made me describe this to you, so maybe I shouldn't duplicate it here. Let's look at the pipeline, indications and studies. So the 2 indications are cancer and infections, addressing unmet medical needs in large patient groups. The blue panel in the middle highlights the high precision cancer treatments without side effects, which is what we find both in animal models and in patients giving our lead compound alpha-1H. So alpha-1H is currently in late phase 2 studies and is being developed further towards phase 3 trials. High precision anti-infectives is the other part. And why are they high precision? Because we have for many years discovered and explored the molecular mechanisms of disease. Why do you get sick when you get a bacterial or a viral infection? What are the defects in the immune system? What are the molecules that decide if you're going to be protected or sick? And how can we find targeted drugs that put things right? And there are 2 examples here. The IL-1 receptor antagonist, which is used for 2 indications. One is bladder pain and the other recurrent cystitis. The bladder pain study, we have performed successful treatment, off-label treatment, and shown that this is working in about 50% of treated patients. And we are now doing a placebo-controlled phase 2 trial to confirm that this is drug-related. The alpha-1RA is also used for patients with acute cystitis, and I will come back to this. In addition, there's a rich pipeline where we leverage groundbreaking IP for several projects and execute our clinical strategy to commercialize the drug portfolio. So first, bladder cancer. Our anti-cancer technology is groundbreaking. There is no compound described with similar effects. I think the efficacy or the mechanism of action is illustrated in this slide, where you see your cells in a red color, which is our drug, and the dotted line highlights what happens. You can see the tumor disintegrating, forming single cells. And in the urinary tract, when the bladder is voided, these tumor cells are just leaving the body. Bladder cancer, why is this essential? Bladder cancer is declared by the FDA an unmet medical need, especially the late BCG-resistant disease. This is the 4th most common malignancy in males in the United States and 5th in Europe. The most expensive cancer indication. Why? Because more than 80% of patients with early bladder cancer recur after complete surgical removal of the first tumor, a very high recurrence rate. and there are few effective treatments. This is why this is an unmet medical need, and where so many are trying to find safe and effective treatments that prevent recurrences and stop disease progression. High mortality rates, of course, in patients who experience invasive disease. And the other side of the coin, a huge and growing market. I will come back to this later. Our alpha-1H, our new bladder cancer drug, is very broadly active, but I will focus now on its effect on bladder cancer. We may come back to other indications later. So the investigational drug is a complex between a protein called alpha-lactalbumin that you see to the right, and oleic acid, a fatty acid. And starting with Hamlet, which was the whole protein, we then developed a synthetic compound based on alpha-1, which is the red peptide that you see in this slide. This forms a complex with broad tumoricidal activity. We have developed GMP manufacturing, of course, otherwise we couldn't do clinical trials. Proven safety and efficacy in animal models of cancer, and therapeutic efficacy, first in a randomized placebo-controlled study of bladder cancer without drug-related side effects, and subsequently in a dose escalation protocol. And as I mentioned, FDA has approved the IND and later given us fast track status. So alpha-1H is helping the company to advance precision cancer therapy. Mark asked me why, what was the discovery? And of course, discovery was the whole Hamlet molecule and its potent effects in patients with bladder cancer. But you can't develop the whole compound as a drug. And this is why we also chose to develop the smaller synthetic peptide alpha-1H variant of Hamlet, which has enabled large-scale production and clinical trials. So the clinical stage is that alpha-1 has successfully passed the phase 1/2 trial with the placebo control and drug escalation studies. And a recent combined data analysis of these two clinical study parts showed a very strong effect. So 82% of the patients showed a reduction in tumor size treated with a higher dose, and 45% of tumors treated with the lower dose showed a reduction in tumor size. And continued clinical trials here are, of course, happening as we speak in dialogue with the FDA. What is the strategy for cancer therapy? So our primary commercialization strategy involves partnering with pharmaceutical companies. And with the successful trials and the fast track status, we think the time is right now for partnering. We are looking both in terms of financing for collaborative studies and or partnering for the conventional drug development structure. This image is to show you a little bit more hands-on what happens with the treated patients. So the bladder, which contains cancer, is usually exposed. This is how patients are treated. They are exposed through a catheter to a drug that is attacking the tumor locally. And there are several drugs that are being used for this purpose, including cytotoxic drugs, BCG, and other treatments that some of you have probably heard of. So alpha-1 is given the same way. It's injected into the bladder to reach the tumor. And when it reaches the tumor, it's rapidly taken up and the patients are shedding pieces of tumor and tumor cells into the urine. This happened within 2 hours of treatment. And you can see just by microscopy that the increase in number of cells is very strong. And also the staining for the substance, the alpha-1, which is inside the tumor. You know that many drugs pose a problem because they do not automatically invade the tumor. Alpha-1 is very invasive for tumor tissue. After the treatment has ended, either the tumor is gone, is smaller, or it's not affected. And this is where our positive statistics are very encouraging. So a reduction in tumor size in the treatment group. Working with the pathologists and the surgeons also makes it possible for us to investigate the molecular changes in the tumor in great detail. We use RNA sequencing, modern omics technology to really define what is going on, how the treatment works. And it's possible by screening for proteins, immune response proteins, to really pinpoint what the tumor is doing when it reduces its size. Some more statistics. To the left, you see the responding tumors, the percent that have a reduction in tumor size. So the placebo group, the lower dose, and the higher dose, and there's this increase to almost 80% responding tumors. Alpha-1 uptake, the red color, where the placebo, of course, does not see any alpha-1, so it's not positive. And there is a dose-dependent increase in uptake by the tumor cells. And finally, apoptosis, because the mechanism of tumor cell death is an apoptosis-like mechanism of death, and the frequency of cells that are apoptotic increases with the dose. To the right, the pictures show the uptake. The red color is the uptake of the drug by the tumor cells, and to the right, the apoptosis inductions in the tumor. So reduction in tumor size and number, uptake of the drug, rapid shedding of tumor cells and apoptotic cell death, inhibition of cancer gene expression, which is very interesting. And what I haven't mentioned so far, but of course is a key, is that these patients do not experience drug-related side effects. So there have been no significant safety issues in this regard so far. So after the placebo-controlled study and dose escalation study, which have been completed, we are now performing repeated treatment protocols to define the protocol for the future phase 3 trials. In summary, cancer treatment with documented clinical efficacy and very low side effects. So who is going to be treated? Hamlet Pharma chose early bladder cancer as the first indication, because previous studies with Hamlet had shown clear effects on this particular form of cancer. Targeted cancer eradication. So our data shows that alpha-1H has unique ability to target and eliminate cancer cells with precision, preserving surrounding healthy tissue, making this a very promising advancement in bladder cancer therapeutics. And as I said, our primary strategy, commercialization strategy, involves out-licensing of alpha-1H to a pharma partner, hopefully, that can take it through the development of phase 3 to market approval. So future strategy with the fast track designation, we would like to take alpha-1H through phase 3 trials in dialogue with the FDA, EMA in Europe, find a suitable commercialization partner, seek FDA approval for market approval, develop alpha-1H for additional cancer indications, and here we return to Marc's question about why bladder cancer. Of course, we are working with other indications, including brain tumors, and we may return to that, Marc, in the questions after the presentation. We also have a very rich pipeline. The market for bladder cancer is huge, since it's one of the most common and most expensive cancers. I chose this slide. It shows the development, the increase in bladder cancer market with time. It's limited to 2025, but I like it because it includes other indications that we are working on, the glioblastoma, colon cancer, and also human papillomavirus, where we have very interesting results from the Hamlet era. So the combined market for alpha-1H, were it to attain its promise, to keep its promise, is of course very, very strong. And using global data, their overview of the bladder cancer market, we see equally huge numbers. You have all seen cancer market numbers, but these are validated through global data. So that's the part about bladder cancer and alpha-1H. I would now like to turn to our two main indications where phase 2 trials are ongoing in the infection-related work. Immunotherapy, a complement to antibiotics. You all know that antibiotic resistance is a huge problem. I don't need to mention that. But why immunotherapy? The intuitive, I think, response to lowering the immune response would be that the patients will become more susceptible to infection and not less. And you vaccinate to improve immunity. So why are we using immunotherapy to inhibit the immune system? Well, it's because when you get sick from severe infections, COVID is a good example, your symptoms and the pathology reflect the fact that you get too much immunity. They call it a cytokine storm, and it's really the case. Your body will explode with an immune response that is not controlled. And you need to return the immune system to a balanced situation where it is able to control, not least, its antibacterial effect so that you get rid of the infection. And this is what we have been able to do. So in phase 2 trials, we are looking at immunotherapy against bladder pain syndrome and recurrent acute cystitis. And this is using a drug that is already approved for hyperinflammatory diseases such as rheumatoid arthritis and other examples, which inhibits an interleukin called interleukin-1. The targeted medical problems, bladder pain affects about 0.1% of the global population. And this is a disease which causes severe debilitating pain in patients who basically have very few treatment alternatives. You can remove the bladder, in some cases that helps, or use morphine and other very strong drugs. But this problem is chronic and very difficult to cope with. Recurrent acute cystitis, a less severe medical condition, but very, very common. Bladder pain syndrome treatment, I mentioned that we have been able to use off-label treatment in this patient group to do a pilot study asking if there was any effect of the immunotherapy. And we actually found very positive results showing relief from severe symptoms, increased quality of life. Some patients could return to work, they could sleep through the night. It was a huge improvement. And laboratory findings, as I show here, confirm the positive effect of treatment. What this immunotherapy does is to target the nerve cells. To the left, you see a nerve cell full of color. The green and the red are the molecules that cause pain, that send signals to the brain that this is a hurtful event in the periphery of the body. When we treat with the nerve cells with immunotherapy, you see the color is gone. There is a very strong anti-pain effect that can be measured, for example, in this panel. What about the anti-infective effect? It turns out that treatment with this inhibitor does both block inflammation and improve bacterial clearance. It's counterintuitive, but the data is very clear in this model. Uninfected bladders, these are mice, obviously, show very delicate structure. Infected, in our model, very strong inflammatory response, red, enlarged. Antibiotics remove part of this response, but the immunotherapy removes both the inflammation and the bacteria. So log CFU in urine, these are bacterial numbers in the untreated, the immunotherapy treated, and the antibiotic treated animals. So this effect on the bacteria is very comparable, and inflammation is reduced more effectively by immunotherapy than by antibiotics. From this, it follows, and I would like you to notice this because this is very important. From this follows that we can also treat antibiotic-resistant bacteria, which is shown here in this slide again, the normal uninfected, the good outcome of immunotherapy, and the fact that the antibiotic cefotaxime used here has no effect. So this is an area in 2024 that we are very excited about and hope to develop further. Is this important? Acute cystitis is one of those diseases that everybody has. Half of all women have it at least once. 160 million women annually are affected by recurrent urinary tract infections. So it doesn't make any sense to talk about market size in this regard, really, because this is a disease where antibiotic resistance is rampant, and there is a huge need for new therapies. I don't even show you, I think, this market size, because I think you can imagine it for yourself. So the future strategy here is different from the alpha-1H, because this is an approved drug with a new indication patent where we are gathering clinical data. So our prime intention is to find a suitable commercialization partner, and we need to discuss the extent to which phase 3 trials are required. At least bladder pain should be an orphan indication. We haven't checked it, but it should be. And of course, seek market approval. And then in the infection area, we have a very rich pipeline of other discoveries. So this pipeline is overviewed here. So you have the two, the alpha-1H for bladder cancer and the IL-1 receptor antagonist, where phase 2 trials are ongoing. Alpha-1H is also being developed for brain tumors. Hamlet, we are looking at whether or not it would be possible to go back and make the whole molecule. And in that case, prime indications would be colorectal cancer and oral cancer. Other pain inhibitory molecules, and something that is very, very exciting, what we call an RNA-APL2 inhibitor. It's enough if you remember that it's a very, very strong anti-inflammatory, general anti-inflammatory substance that we actually have recovered from nice bacteria. We also have an inhibitor of bacterial infections of the kidney and urosepsis, and tuberculosis peptide that works also against resistant Mycobacterium tuberculosis strains. So a company with a lot of potential. We are careful to focus and restrict our activities, clinical activities, to these 3 indications, where we hope to have more good news in the next year. But there is also, I don't know, it's a bit like a candy store. We have very good publications for the new substances that we also would like to develop. So I would like to thank everyone who's been involved in the work of Hamlet Biopharma and the academic work that has led to these projects being possible. And thank you for your attention.
Great. Thank you, Professor Catharina Svanborg. Thank you for sharing Hamlet's exciting, novel, innovative therapeutics pipeline. If anyone has any questions and or interest in Hamlet, you can contact us here at MSQ or connect. We can connect you surely with Professor Catharina Svanborg and management. But now we're going to collect some questions from the audience. If you give us a couple of minutes. So here is one, Professor. Can you elaborate on the unique properties of alpha-1H and how it distinguishes itself from the existing treatments for bladder cancer and brain cancer?
Thank you very much. Yes, alpha-1H has a combination of properties that is different from most current treatments. It kills tumor cells, obviously, very, very effectively, and it also lacks severe side effects. In our placebo-controlled study, there were no drug-related side effects recorded, and we can see a bit of local irritation if we increase the dose, but nothing that is very significant in terms of side effects of cancer therapy. So we believe with this combination that we can target early bladder cancer. We know that there is always a lag. In most countries, there is a lag between the time of diagnosis and the time of surgery. And at the present time, there is really no therapy offered to the patients during this time. We are seeing alpha-1H as a drug for this period, potentially delaying the need for surgery. there is obviously also a potential for treatment of recurrences after the primary surgery. So we are optimistic about this.
Great. Here's another one. How do you foresee alpha-1H fitting into the current treatment paradigm of bladder cancer and brain cancer? Any plans to explore a combination therapy?
Definitely. We are regarding alpha-1H as a neoadjuvant. That's the position that we are hoping to take for alpha-1H, and indicating that the patient will need additional therapies, as always in cancer. And we have very exciting data that I can't go into in full, but suggests that there is good reason in terms of the immune response to expect synergy with some of the known therapies. So we are looking at that further.
Great. Would proactive or post-surgical treatment with alpha-1, when considering tumor resection surgery, improve the effectiveness to prevent GBM recurrence?
This glioblastoma,
Yes.
GBM, so we are leaving bladder cancer, moving into the brain.
Correct. Yes.
Yes.
That was the question.
Yes. Okay. So the work done in brain tumors was done in a xenograft model where primary human tumors obtained at surgery were first cultured for a few days in the lab and then injected into the brain of immunodeficient rats. This is a model developed by Professor Björkvik and his group in Bergen, Norway, and they are internationally very well established. So the tumors grow from the point of injection. And we were able to infuse, by convection enhanced delivery, infuse Hamlet into the brains of these rats for 24 hours after 7 days, and then leave the animals to develop tumors. And after about 8 weeks, the controls were sacrificed, and there were large tumors at MRI scans. At that time, tumors in the Hamlet treated were very small. This is the basis for going on, so to say. We also have very good responses in the lab for a variety of human brain tumor cell lines. We haven't found anyone that is resistant so far. The key to this collaboration now, why we have picked up on the brain tumors again, is Professor Gill in Bristol in the UK, who is a world-renowned expert in the field of brain tumors. He's a pediatric neurosurgeon and has developed platforms that you can use for convection enhanced delivery into the brain. So the current collaboration aims to, with him and his group, aims to define a combination of an injection platform and alpha-1. And in view of the efficiency in bladder cancer and different technical details that Professor Gill is solving, we hope that this will be really fruitful.
Great, thank you. Couple more. Could you share Hamlet's research and insights into the blood-brain barrier penetration of alpha-1H?
This is a key question, isn't it, for brain tumor treatment? And so many have struggled with this. I think we have no claim to fame in this area ourselves, but working with Professor Gill, who has developed these devices for direct injection of the drug, bypassing the blood-brain barrier, is really a key. And I think it has already been used for other types of indications of brain-related diseases with success.
Great. Here's another one. Are there any efforts to identify specific biomarkers or patient characteristics that might indicate a particularly favorable response to alpha-1H? And how might this influence treatment strategies?
This is a key question and one that interests us a lot, obviously. There is something remarkable about alpha-1H, which is that we haven't found any patient that doesn't respond yet. Doesn't mean that all patients respond to the same extent, but just looking in the urine at cell shedding, we find in the patients who are treated with this higher dose that there is shedding in all of them at some point during this 1 month period. So the question is if cell shedding is the biomarker. We are also with RNA sequencing, which is very, very interesting, finding some extremely strongly regulated genes. And the key then is to see that regulation early enough to use it as a predictor. The third variant is the immune response, which also is very rapid and extremely strong for some markers. In all of these cases, the biomarker appears after the first injection of alpha-1H. So the question is whether a design should be to first give a patient one or two injections and then look at the response and predict future efficacy based on that. That would be a possibility.
Great. Great. I think that's it for the questions. So congratulations, Professor Catharina Svanborg, on your discovery and all your accomplishments to date. We're all excited and eager to see Hamlet's future milestone achievements going into the new year. Any last words you want to share before we conclude?
I think it's exciting that we have been able to reach this point, and we're also very motivated to partner to make sure that these discoveries are developed further and reach the patients in the market.
Great. Well, thank you. Thank you, everyone, for your interest today in Hamlet and in MSQ CEO Founder Webinar Series. Have a great day, night, happy holidays, and early Happy New Year. Thank you.